Assessment is very important too. The matter of transport time and transport conditions, of course, because we have a problem with things dying or degrading, or simply because the human body is certainly not a sterile site, there are a lot of contaminated and native organisms present. If we're trying to assess how the things are swimming, we want them to be swimming. We don't want them to be half dead and tired and worn out. It's asked for them to be refrigerated to try and stop overgrowth, but that doesn't always occur. And also some things like this, which is called a cast, break down really fast. If you think about sausages, cast is basically like a sausage. The kidney consists of a whole load of little tubes, and if you force a fairly solid thing through a tube at pressure, it's going to make a sausage. So if the urine is too high in protein, for example, or if it's got a lot of cells up at the kidney stage, then it's going to make these little sausages, and they're called casts. But because that sausage doesn't actually have skin on it, shaking it up or just being around too long, it's going to dissolve out into the remaining solution. And you can see that one there is almost invisible. Crystals will tend to precipitate out over time, and there's not too many there. You could still see cells are present. If you can imagine if you get a lot of them, you won't be able to see how many cells are present. We also look at the type of cells present. If there are lots of white cells present, that indicates the body's reacting to something, so it's much more likely to be an infection and contamination. If there's a lot of epithelial cells present, that means there's a lot of cells from the outside of the body in the sample, which means there's also more likely to be contamination. So if we have bacteria in conjunction with white cells, we will tend to report it as an infection. If we have bacteria in conjunction with just epithelial cells, it should be contaminant. But if it's got both, we tend to say we cannot prove it's an infection because it could just be a contaminant. Epithelial is the skin cells. It's been contaminated from the skin on the outside because it's not really a true midstream urine. There can also be epithelial cells from the bladder itself. They're quite a different structure, and we can tell them apart.